Hello everyone, and welcome back to AP Bio with AP Playground. Today we're covering topic 2.9, cell compartmentalization. Objectives today are pretty simple. We're basically focusing on describing the membrane-bound structures of the eukaryotic cell, specifically the organelles, and especially how this creates compartmentalization, which separates intercellular metabolic processes and specific enzymatic reactions. And our suggested skill is going to be argumentation 6E, predict the causes or effects of a change in or disruption to one or more components in a biological system. Firstly, separation of reactions. Different reactions are going to take place in different environments where they can happen the most efficiently. This is known as compartmentalization. And within eukaryotic cells, this is going to happen due to internal membranes that each organelle is going to possess within the cell. So why is this important? First of all, it allows catabolic and anabolic reactions to be separated, the making and breaking reactions. Second of all, it allows for each enzyme and function of the cell to happen as efficiently as possible. Because first of all, you're keeping it separate from the rest of the cell, and second of all, you're allowing it to have its ideal range of temperature, ideal range of pH, and ideal range of salinity in order to function as best as possible when you keep it enclosed within its own membrane. So if you look over here in this diagram, this is the pH of subcellular components. And most of them have a pH of around 7 because that's kind of the base for the human body. But a mitochondria's got a slightly higher pH, and the Golgi network has a slightly lower one. So every single organelle is going to have its own optimal pH, and that's why it's so important that it's kept within its own membrane so that the pH doesn't affect the rest of the cell. And you can even see this probably the best with the lysosome, which is basically a ball of acid that helps with digestion processes, basically breaks old things apart or unneeded things. And if the lysosome membrane were to break open, the entire cell would be engulfed in this acid and wouldn't survive because the pH is so low. So that's just the importance of having the membrane enclosed organelles. And this compartmentalization is going to apply to the whole body. We don't have stomach acid everywhere in our body because our body can't tolerate it. It tolerates it specifically in the stomach. It's going to have its own pH, and processes are separate so that digestion is only going to happen within our stomach. Secondly, compartmentalization can help increase surface area. Like we said, in eukaryotic cells, compartmentalization happens due to the membranes that each of the organelles have. And we can see this most clearly with mitochondria and chloroplasts, which have double membranes, meaning they've got an outer membrane and an inner membrane. And also the inner membrane is folded up, which means there's a huge increase in surface area. That means more resources are able to be brought in and expelled, and there's a larger production of energy, especially when we're looking at the mitochondria. Ion gradients can also be established using the inner membrane, and it's not going to affect the rest of the cell. So there's going to be travel of hydrogen ions across this inner membrane, and you'll learn about that when going over cellular respiration, but it's not going to affect the rest of the cell in terms of pH levels, because of course hydrogen ions are going to increase or lower pH because of this outer membrane that the mitochondria has. So membranes really allow for separation of processes, and they allow for a larger area in which to create processes. We get more resources in and out of each of the organelles, and the cell becomes more efficient overall. Lastly, let's talk about the means of compartmentalization. So mitochondria and chloroplasts have their own DNA, which means they can regulate their internal environments without the assistance of the nucleus. They're able to determine what specific proteins they need made in order to function, and they obviously also have their own ribosomes. Proteins created in different parts of the cell get transported to different areas based on where they're created. So if you're looking at free versus bound ribosomes on the endoplasmic reticulum, proteins created in the cytosol and free ribosomes usually stay in the cytosol, while those created on the endoplasmic reticulum generally get exported out of the cell. And lastly, cell division keeps compartmentalization because it keeps all of these membrane-bound organelles and duplicates them when creation of a new cell happens. So, review compartmentalization. Know why it exists for the separation of reactions and the increase of surface area, and we'll talk about its origins in the next lesson.